Hey, so I'm Victoria Rattel, and today my colleagues and I are going to talk to you about when viruses attack in the Gulf of Mexico, specifically looking at lysogeny in the hypoxic dead zone off the coast of Louisiana.
So, hypoxic or oxygen minimum zones refer to the conditions in water that have oxygen concentrations so low that little to no macroorganisms can live there. Scientifically, hypoxic regions have an oxygen concentration less than 2 to 3 milligrams per liter. The map shows oxygen concentrations around the world. Oxygen minimum zones are created when oxygen in the water is being consumed by heterotrophs faster than oxygen is being produced. When water isn't circulating, that means there's no mixing. Some more bad news is oxygen minimum zones are growing in relation to excessive runoff. Secondly, currently the Gulf of Mexico is described as being a seasonal hypoxic region. In the summer, ice begins to melt and watershed channels excessive nutrients from runoff into the warm Gulf of Mexico by the Mississippi River. The map shows where the runoff is coming from. The runoff causes an algal bloom in the Gulf. When the bloom sinks, bacteria and heterotrophs decompose the phytoplankton and the algae and consume the oxygen. This causes the hypoxic regions in the Gulf. This is bad because the macroorganisms leave the area, meaning there's no seafood for the seafood industry to use. Virus-host interactions. There are two types of interactions. First is the lytic cycle. The virus meets its host, injects its DNA, replicates, then releases from the host. This strategy can be seen at the top diagram. The second interaction is the lysogenic cycle. This strategy, the virus has a choice. It can go straight to the lytic strategy and kill its host, or it can integrate its genome into the host genome and stay there until triggered to switch to the lytic strategy. The cycles depend on different locations of the ocean. A theory is that areas with sudden increases of productivity, like seasonal hypoxic zones, will favor viruses that are lysogenic. There is an entire class of genes that viruses have called auxiliary metabolic genes, or AMGs, which is a viral gene not directly involved in replication. The viruses steal these genes and use it to enhance their ability to kill things, but they do not have all the genes that code for each ability because they're too small. Instead, they have a rate-limiting gene that is used to enhance its fitness. The picture shows an AMG with a photosynthetic rate-limiting gene.
So why is all of this important? We can better understand the interactions between microorganisms and anthropogenic-induced hypoxic regions. Shown here, you can see that phosphorus and nitrogen sources are in the Gulf is coming mostly from agricultural runoff. And we can look into the dynamics of viral life cycles, which you can see in the photo below. Further, we can explore oxygen stress in bacteria and see how viruses can play a role. And we can do further research into AMGs, viral AMGs that infect heterotrophic bacteria. And we might see that AMGs can help reduce the impact of hypoxia in the Gulf by bioengineering with viral AMGs.
So we want to investigate the role of viral life cycles in the death of heterotrophic bacteria off of the coast of Louisiana in this dead zone. We believe that with a decrease in the concentration of oxygen and therefore a decrease in heterotrophic bacteria, that viral abundance will increase, which is largely due to the induction of lysogenic viruses into the lytic cycle based off of oxygen stress. So that we should see the highest lysogenic induction during the onset of hypoxia. To confirm this, we also want to see if we have viral genes for lysogeny that should be present in all of our samples, but expressed at higher rates during the onset of hypoxia. To start out our methods, we need to collect samples. So off the coast of Louisiana, there are 4 highlighted areas in which we'd like to collect samples over a 2-year period. The samples will be collected before, during, and after the onset of hypoxia at a depth of 30 meters to avoid the surface where there's photosynthetic algae. The dissolved oxygen concentrations will also be measured, and then some samples will be set aside for metagenomics and metatranscriptomics, while the others will be filtered, stained, and counted.
When it comes to finding viral abundance and percent lysogeny, we can use epifluorescence microscopy to determine the viral population. We would take the other half of our filtered samples and stain bacteria with cyber gold to allow us to easily count them later and determine the total abundance. From there, we will introduce mitomycin C into the samples to induce viral replication, acting as our control sample. We can then use the same microscope to determine the percent of viruses undergoing lysogeny. This will show us the percent change in each viral cycle when hypoxia occurs. We will also count the infected bacteria using FBIC and a transmission electron microscope to double check we have inserted viruses into the sample. The methods for finding metagenomics and metatranscriptomics start out very similar to the methods for counting. However, all these steps should be done under an anoxic hood to preserve the levels of dissolved oxygen to keep our genes exhibiting the same expression. We will sequence the extracellular viruses and once again add mitomycin C to half of the samples to act as our control. We could then insert both sequences into virus order, and we're hoping to find that an increase in genes will occur when the bacteria are undergoing lysogeny, and it will act as a potential quantitative measure. We would then use metatranscriptomics to see the expression of genes in our samples, allowing us to compare the abundance of their expression during hypoxia.
When we analyze our data, we're going to want to see some results in order to support the hypotheses that we put in place previously. In our samples, we're going to want to see a peak in the amount of viruses when the population of heterotrophic bacteria begin to decline. We should also see a large percentage of lysogenic viruses being measured by increase in viruses after the addition of mitomycin C. Genes associated with lysogeny will be present and expressed at higher rates during the onset of hypoxia indicated with our metatranscriptomics data. If the mitomycin C induced samples have higher lysogeny, it's possible that the lysogenic viruses have genes to deal with oxygen stress in hypoxic regions. If you have any questions, please feel free to email any of us. Thank you.